So yeah, I'm working in the field of chemical ecology, and not everybody of you may be familiar with what we do in chemical ecology. So what our main focus is, that we are really interested, in our group at least, in interactions between plants and herbivores. And here we want to know how the plant chemical compounds affect herbivores in their performance, but also in their behavior, in their personality. So different aspects of effects that they can have on the insect side, but then we are also interested how the insects actually can affect the plants. And traditionally, chemical ecologists mostly looked at characteristic key compounds. So as Eva mentioned, the glucosinolates for brassicaceae, or here the iridoid glucosides for plantago. But within the last, I would say, 10 to 15 years, we are looking now more and more also in overall metabolic fingerprints. So how is the overall chemistry changing in response to certain abiotic or biotic factors? And of course, these above ground interactions on which we mostly focus are also fine-tuned by below ground interactions, because you may have mycorrhizal fungi, for example, living in symbiosis with the plant, and due to this nutrient exchange between the plant and the fungus, you may also expect that above ground the chemistry changes and then can affect above ground interactions. And in addition, you may have also different abiotic conditions like fertilizer, like water availability, or heavy metal contaminations that then can affect plant chemistry and can affect these plant-insect interactions. So these environmental stressors are something that we are getting more and more interested, and of course we all know that currently our ecosystems have to face different global change aspects. So it could be drought, that is also studied here very well in the DRESS project, but it's also temperature changes. So last year was the hottest year in Europe, and we had all these new records of highest temperatures at different places throughout Europe, also in Germany. And then of course ultraviolet radiation is also increasing and changes a lot also plant chemistry and plant-insect interactions. Then we have plant invasions as a very different aspect of global change, where we can also have interesting hypotheses on how chemical changes affect plant invasions. And we have all these different aspects that have to do with increasing world populations, increasing use of agrochemicals that change plants and herbivores, and also contaminations of the soil or of the air that have strong effects. And so we study different aspects in this global change effects, and today I just want to focus on 3 aspects, on droughts, invasions, and contamination. So quite different topics, but I hope if you do not find one so interesting, maybe the other one is more interesting for you, so you can sleep at some time and then wake up again at the other time. But these are the topics I want to focus today on. So let's start with drought. For droughts, often, so in laboratory experiments, people just reduce the amount of water and compare that with the control group. But then, of course, we should not only think about reduced water, but we should also think about different watering events. So we may have periods due to global change where we have no rain at all, and then we may get all of a sudden a lot of rain. And that's what we try to test. So not only have reduced drought, but also this pulsed drought events, as we call it. And we studied this in a crop plant, actually, in wheat. So we had in our wheat plants, control plants, normally watered, then continuous drought. So they are also watered every other day, but with less water. And then we had a third treatment. Here the plants only got water every 8 days, and we call that pulse drought, but they get then quite a lot of water, namely all the amount the continuous drought treatment got over the time. So that should affect, of course, the plant, and you would expect that under drought conditions the plant does not produce that much. But in nature, there is not only the plant and water and some nutrients, but you may have also symbionts in the soil. So you may have mycorrhiza. And mycorrhiza is actually very important, because if the plants are mycorrhized, then the roots can get better access to not only phosphate, but also to water. So drought effects may be reduced due to mycorrhiza that improves the water availability for the plants. And due to this nutrient exchange, so the mycorrhiza gives the plant more phosphorus, and in turn it gets more photoassimilates. The above ground chemistry changes, and that in turn may affect then, for example, aphids sucking on the phloem sap, where we have the transport of nutrients from above ground to below ground, or in both directions. So it has many factors, our setup. So we had these plants either not mycorrhized or mycorrhized. And then at some point we put aphids in clip cages on the plants. And in the end, the plants were harvested. They were under these 3 drought treatments, as I just explained. And then we looked at the above ground biomass, the biomass of the ears, so the reproductive parts. And we also looked at the phloem sap chemistry, because that is relevant then for the aphids, particularly because they only suck on the phloem, so it doesn't make sense to look at the overall leaf chemistry if you're interested in how aphids respond. So just a picture how that looked like. So as you would expect, under control conditions, plants grow very rigorously. If they have continuous drought, they produce less biomass, and under pulse drought, they produce even less biomass, and they look already a little bit dry. But what was for us very interesting, that actually if you look then how much the plant invests in the overall biomass and in relation to the biomass that is invested into the ears, so in the reproductive parts, then the pattern looks a bit different. So these are first the data just for the non-mycorrhized plants, control conditions, continuous drought, and then pulse drought. And what you see is actually that under a little bit of drought that is not too harsh, actually the plants invest even a little bit more in the ears compared to the overall biomass. So from agricultural aspect, actually a little bit drought isn't too bad, but then if you have really this pulse drought events, then it gets pretty bad, because then the plants also start investing much less in their reproductive parts, and the harvest in the end is also pretty low. What was now interesting is if we have mycorrhiza, then actually the plants under control conditions had the highest degree of mycorrhization, so the best association with the fungus, whereas under these drought conditions we have only 10 to 20% of mycorrhiza. That is not that much, but overall in grasses we always have much less mycorrhiza than we have in herbs. Yeah, in herbs we have 80 to 90% total root length colonization. In grasses we always have less. But even though these are even less mycorrhized, we get actually an improvement. So as soon as the plants are mycorrhized, actually, at least under the drought conditions, they invest proportionally more into the reproductive parts compared to the vegetative parts. So for agricultural systems, it means that it's maybe it's good not to use any fungicides and not to kill the fungus, because fungus can be very beneficial in that case to improve the crop in the end. And of course, if we have these effects on the plant biomass, then we should think also about the acid composition in the phloem sap, and the amino acids are very important for the aphid digestion. So here again, it looks a bit busy, but here we have the control plants, the continuous drought and the pulse drought, always non-mycorrhized and mycorrhized. It's very busy and colorful, but I just want to point out 2 amino acids that show up here and are very important. One is the purple one here on top. So you see under drought, stress, proline, which is a typical osmolyte that is important to keep up the turgor pressure under drought conditions, increases under drought conditions. But interestingly, once the plants are mycorrhized, you do not see this drought signal anymore, because then probably these plants are not that drought stressed anymore, as the mycorrhiza helps to get access to water that is still available in the pots. And then another amino acid that was quite interesting is glutamic acid. And here for glutamic acid, you also see, particularly under drought conditions, non-mycorrhized and mycorrhized plants, that in the mycorrhized plants we have an increased amount of glutamic acid, or increased relative proportion of glutamic acid. And glutamic acid is important for nitrogen transfer in the plants. And of course, when the plants are mycorrhized, then there may be also increased nitrogen transfer. So that's how we interpret this data. So if you see now already, okay, drought and mycorrhiza have an effect on the amino acid composition, then you can also expect that that has an effect on aphid performance on these plants. And it is well known that slight stress can have actually a positive effect on aphids, because For example, proline is also quite important for aphids, and it also affects the turgor pressure of the plants, and aphids depend not only on the chemical composition but also on turgor pressure. So indeed what we found is not significant differences, only tendencies that under continuous drought aphids perform a little bit better. So in the beginning we placed 20 aphids on all plants, and then 14 days later we look how many aphids are still on the plants. But you see under pulse drought stress conditions, plants have so little turgor pressure that aphids just basically cannot survive on these plants, which is maybe also what you would expect. Now, if we look at the mycorrhized plants for control continuous drought and pulse drought, you see that actually mycorrhiza makes it worse for the aphids, better for the plants, because then they are less infested by aphids. So long droughts and obviously also mycorrhiza can impair aphid performance quite drastically. And of course, that is not only due to amino acids and turgor pressure, which is important for the aphids, but then a lot of metabolites, additional metabolites, may change due to the mycorrhiza. And a couple of years ago we did a review and looked at all different compounds that may be affected by mycorrhiza, and we know that a lot of primary metabolites change, so the ones that are common throughout all plant species like fructose, glucose are all increased due to mycorrhiza. Several organic acids are increased, but some are also decreased due to mycorrhiza depending on the species. And then we have a lot of secondary metabolites that may respond in different directions. They may increase or decrease. But all these effects have been mostly looked at the overall leaf level. But in the phloem sap it may be completely different, because in the phloem you do not find all these metabolites, and only very few people really look at the phloem sap composition. So that is something we are trying to build up now more and more. So just to sum up this first part of my talk, we see that both the intensity but also the frequency of drought events can be very important if we look at effects of droughts. For the aphids, of course, we really need to look at the phloem sap, and I will come back to aphids later on, because somehow they are getting now my new pets. And then, yeah, for the wheat itself, we could say that maybe the mycorrhized wheat are a little bit happier, at least under drought conditions, because they then invest relatively more into their ears, and they are less affected by aphids. So that has indeed an advantage if we have mycorrhized wheat. Okay, yeah, switching to a completely different topic, but I think plant invasions are also something that intrigued ecologists already for many decades, and some of you also worked on plant invasions, like Eva, for example. And here the question always is, why do plants actually get so successful in the new environment, although they were never really problematic in their natural habitats? Yeah, and there are couple of hypotheses that also have to do with chemical ecology. And so for these plant invasions, so these are plants that are, for example, native in North America and then got introduced to Europe and start becoming invasive there. Actually, the other way around, it happened much more often, but just to have that as an example. So we have transcontinental changes, but also within a continent, we can have movement of plants in a newer area and then get to plant invasions. And I will bring two examples, one of a transcontinental and one of an intracontinental range expander. So for successful plant invasions, we may have different ideas. So one could be advantageous pre-adaptation. So the plants may just produce a lot of seeds, or may have a very efficient vegetative reproduction that makes them very successful. But we could also have some post-introduction evolution. So once they are in the new area, they may select, or selection may be driving some traits in certain directions, okay? And that all, of course, depends on the genetic architecture of the plants. And there are some ideas, hypotheses that are related then particularly to chemical ecology. And one idea is that in the new area where the plants come to, that they are lacking enemies, natural enemies. So the plant is introduced, but maybe not necessarily the natural enemies. So that may cause a shift in the chemical defenses, because if I do not have that many enemies, then I do not need to invest that much in defense, and I can invest instead in growth, and that may make me more competitive. This is one idea. But then we have to consider that there are different types of chemical defenses. So it gets a bit more complicated because we have, on the one hand, qualitative defenses. So qualitative defenses are very cheap compounds, cheap to produce. They are very effective against generalists, but for specialists, they would be attractive. So actually, in the invasive range, we still may have some generalists, but we don't have any specialists anymore. So here we may have really the case that actually qualitative defenses may go up in the invasive range, not down as originally expected. And then on the other hand, we have quantitative defenses. So these are ones that are quite expensive to produce. They act on both, on specialists and on generalists. They often act as digestibility reducers. And if we have less herbivory overall, then plants should not invest that much anymore in these expensive defenses and can invest instead more into growth. Okay, so keep in mind that these are two different kinds of classes, but it's not always so easy to say whether it's a qualitative or quantitative defense. And then a third concept here also in context of chemical ecology and plant invasions are novel weapons. So the idea is that plants have some chemicals that they bring into the new environment that the new environment is not adapted to. So these are compounds that the new environment doesn't know, so they don't know how to deal with it. So we were interested in all these three ideas in two different systems, as I said, and the first one is Buddleia davidii. I don't know whether it also occurs here a lot. It's an ornamental plant that people like because it attracts a lot of butterflies. The flowers attract a lot of butterflies, and for that reason it was also introduced from its native area in Asia to Europe, and then also to North America, and now it occurs almost worldwide. And that plant, indeed, if you look in Asian populations where it is native, then you see, at least on the leaves, that there is much more herbivory. But if you look in Europe on the leaves, there's almost no insect herbivory. So the flowers are, of course, visited by insects. But if you look at herbivore damage on the leaves, there's almost nothing. So enemy release seems to be true. Yeah. So there's support for these data. But we were now more interested also in the chemical differences between these populations. So we got our hands on 10 populations from the native area and 10 from the invasive area. And colleagues in Halle grew these plants in a common garden. And then we came and just harvested these plants at one point in summer and got really to the metabolic fingerprints. So had these 200 plant samples in and analyzed their overall chemical composition first. And these data are now plotted in a principal component analysis. So overall, we had almost 2,000 compounds in each sample. And each dot here represents one plant sample with its typical qualitative and quantitative composition of chemicals. And the closer two dots are, the more similar their chemistry is. Yeah, so this plant is very different chemically, for example, from this plant. And then all plants belonging to one population are within these squares. And you see immediately that all the Asian populations are actually quite nicely separated from all the European populations, almost all of them. Yeah, so there's a clear chemical difference, except for this one population, this one Asian population that is just in the middle of the European populations. And that was for us first very confusing, so we thought maybe we mixed up something, but it was really the way it was. And then we looked in the literature and we found that actually originally plants were taken from this Muping Zeng population in Asia and from there introduced to Kew Garden in England and from there spread throughout Europe. So it could be two ways around. It could be that these plants had already a good pre-adaptation and they were maybe had a good cold hardiness or low herbivory, and that was for whatever reason the reason why people introduced it from that population. Or yeah, it could be also, of course, other reasons, but I think it's very nice that you really can, from this chemical fingerprints, we didn't know beforehand this introduction pathway, but from the chemical fingerprints we could predict from where plants were introduced. Yeah. So that is one beauty of metabolomics, even if you don't know what the compounds are behind that. But of course, we also wanted to know a little bit more about the compounds and their role of the compounds. So we did some bioassays with 2 generalist herbivores, one generalist that is feeding on tree species and one generalist that usually feeds on herb species, so Amata mogadorensis and Lymantria dispar. And we offered them leaves of blackberry that they readily feed on. And then we treated the leaves either just with a solvent control or with an extract of Buddleia of our plants. And these extracts were different fractions then actually of the extract. And then we looked at the feeding deterrent activity of these compounds. And we also tested the generalist fungus because, I mean, fungus may have different preferences for different plant compounds. So, what we did is we had a methanolic extract and then we collected different fractions that had different compounds in it, basically, and then we tested first this first fraction in our feeding, and that had no effect, so insects would feed more or less the same amount on control and test leaves, so there's no feeding deterrent or feeding stimulating activity. Then for this second fraction here, actually at least Lymantria dispar was highly deterred, so they fed much more on the control leaves than the leaves just treated with Buddleia extract. And in the third fraction, actually both herbivores responded strongly to that fraction and fed much less on leaf just treated with these extracts or these parts of the extracts from the Buddleia leaves. So that what we can learn from that is that different compounds have different activity against different herbivores. So some herbivores seem to be more sensitive than others. And what is also important here, this first fraction had no activity to the herbivores, but when we tested it to the fungus, actually the fungus couldn't grow on this fraction. It could also not grow on the last fraction, but it could grow very well on that fraction. So also for fungi and for herbivores, different compounds act differently, and that's also important to keep in mind. So if you are chemically highly diverse, then it may also help you to be well defended against all different sorts of herbivores and fungi and pathogens that may be around. So chemical diversity, I think, is a very important feature that is not considered enough. We could identify some of the compounds in these extracts. And so, for example, orcobin and chlorogenic acid are actually known to have also antifungal activity. So those compounds, but also, of course, other unknown compounds here may have had the effect against the fungi. And then verbascoside is a compound that is quite common in European herbs, but not that much in European shrubs. And linearin is a flavonoid that is actually rather rare in European herbs. It's very common in Asian herbs. So it may be something like a little bit a novel weapon, not entirely novel, but something herbivores here are not facing a lot. And particularly all these verbascoside and linearin are really not known from trees and shrubs in Europe. So again, an argument that maybe this is kind of a novel weapon, a shrub or tree that has compounds that are only known for herbivores, but not necessarily for tree or shrub herbivores. We also looked at the intensities in the European and the Asian populations, and it turned out that this maybe more cheap qualitative defenses like orcobin and verbascoside were indeed higher in plants of the European populations, and chlorogenic acid and linearin, which can be considered maybe more as quantitative defenses, were a little bit reduced in the European populations. So that would hint to the shifting defense hypothesis, but then in the beginning I showed you that this one Asian population actually was very similar to this other European population, so it's hard to say whether it's shifting defense or whether it's pre-adaptations, so it's often more complex. But I think we have some good indications for pre-adaptations, we have good indications for enemy release, maybe some indications for shifting defense and some indications for novel weapons. So all these explanations are not mutually exclusive, but they all can actually apply. To switch to another system which I find intriguing, and I also have to talk about brassicaceae. So this is a brassicaceae that is invasive in parts of Germany, for example. It is native to the Caucasus region, but it now spreads more and more westwards within Europe. And it grows on roadsides, but also I saw it a lot growing around crop fields. So if you have an oilseed crop field, then Bunyas orientalis is also growing there. And it has some traits that are typical traits that you often find in invasive plants, such as high outcrossing but also self-healing potential. It has a very high seed output, and it can effectively regrow. If you cut it, it will easily regrow, and it also likes to grow on very fertilized sites. So it has some characteristics that are typical for ideal weeds. And as a brassicaceae, it has this group of compounds that are these glucosinolates Eva already mentioned, that have this sharp taste that you know from all brassicas, basically. And so we were interested in the link also between the chemical diversity, but then also in relation to the genetic diversity. And we wanted to know what are the performance traits of these plants in connection to resource availability, because we know that these plants grow particularly well at well-fertilized sites. So we had plants from the Caucasus region where it is native, also from Turkey, from some populations, and from Romania. Then it is invasive in parts of Germany and also in Lithuania, and it is naturalized. So that means that is a third category. So it's spreading westwards, and there in these populations it's not invasive yet. It occurs, it's not native, but it's also not problematic. Yeah. So we have this third category where we call them naturalized or exotic. Some people also say so in the Netherlands and in France. It occurs, but not that terribly a lot now. So first, what we got was some genetic data together with collaboration partners from Heidelberg. We mapped the genetic diversity, and this is very similar as the PCA I explained before. So the closer two dots are, or the end of these stripes now, the closer they are genetically, and always the mean of a population is now here indicated with a symbol. And you see these three populations, which are these three populations, so these three Turkish populations, they are genetically completely distant from all the others. They are the same species, but they are obviously spread apart already millions of years, or 2 million years ago. So these are genetically quite distinct, and probably source populations for these naturalized and invasive ones were not the Turkish ones, but rather the Romanian or the Caucasus ones. Yeah, so because they lay there. So again, we can say a little bit about the invasion history already based on this genetic data. We also got the metabolomics data for exactly the same plant individuals, and that matches beautifully. So again, we see this separation. The Turkish populations are chemically also quite distinct from all the rest. And these naturalized populations had actually a very low chemical diversity, so maybe some bottlenecks or genetic depletion here in these naturalized populations by causing their lower chemical diversity. And then we just focused on the glucosinolates. So there occur 10 different glucosinolates in Bunyas orientalis, which we really could identify. And here still you see that the same pattern. The Turkish populations are a bit different from all the others. The Turkish ones are native populations. And then we did some feeding assays with a generalist herbivore, offered the generalist herbivore plants from all these different populations. And what we found is actually that the generalist herbivores had their poorest performance on these native populations. So these native populations seem to be very well defended against this generalist herbivore, whereas this invasive and naturalized populations somehow maybe lost a little bit of their chemical defense potential. Maybe it's not needed that much because they may face a little bit less herbivore pressure than these guys here. So to know now, this is all laboratory studies first, but we also wanted to know how these plants actually grow in the field, how well they do if we grow them under common garden conditions. So we set up a common garden experiment, and with different plots. In each plot we had plants of all 12 populations, 4 per group, so naturalized, invasive, and native. And then we had half of the plots well fertilized and the other half not that well fertilized, to also see how they cope with the fertilization. And then we had to wait for 2 years because Bunyas only starts flowering in the second year. And Over the time, we measured a lot of different traits, and we were interested how would the plants perform and how would they respond to the fertilization. So the traits that we measured were a couple of traits related to growth, to defense, to resistance, and then in the second year also to reproduction. And then you have lots of data, and we are thinking about what can you do with all the data. So we thought, okay, maybe first check how much these data actually correlate. So which of these traits correlate strongly, which correlate less. So red always indicates a high negative correlation, so indicating some trade-off. For example, in glucosinolate concentration and leaf mass per area, there may be a negative correlation. But other traits that are indicated in blue have some positive correlations. So for example, glucosinolate concentrations and glucosinolate diversity were highly positively correlated. And then we thought, well, maybe we can use this concept of syndromes that you know probably all from pollination syndromes. So there are plants that have similar traits. They all show red flowers that do not smell that much, or white flowers that smell a lot. So you make groups and talk about different plant syndromes. And we could also do that within a species and look for plants that share certain traits that are more associated and that may also explain part of the invasion success. So we called them then invasion syndromes. So when we look at that matrix and then get some clustering, then we found that actually there are certain traits that cluster more strongly. And these traits were then divided in 4 groups, and I just want to show you data from 2 of these syndromes, as we call them. So let's first focus on syndrome B. So here we have traits that correlate, that are longest leaf and number of herbivores, and then also number of predators and pathogens. Here's always the rank for these suite of traits for the native populations, the invasive ones and the naturalized ones, and always the right bar is the one for the non-fertilized, no, the other way around. The left bar is for the non-fertilized, the right bar is for the fertilized. So the darker one is for the fertilized. And what you see here is what is important. The ranks are always higher here for the invasive and naturalized, so the non-native populations. And these non-native populations had overall longer leaves that were more heavily infested by herbivores. So obviously they just put a lot into biomass and don't care so much about effective plant defense, because they just produce a lot of biomass. With that, they can also grow a little bit more vigorously and are probably also more competitive. And they seem to invest less in costly defenses. So we really have an effect of status that shows us that there is some post-introduction evolution going on. And we have a slight effect of fertilization. So always when they are more fertilized, they also produce more biomass. That's what you would expect. So they can deal well with more fertilization. In the syndrome C, it's a very different pattern. So here we have a clear negative correlation between leaf mass per area and glucosinolate concentrations. So there seems to be either plants that defend themselves well mechanically or chemically, but not both, by investing in both. And also the ones that have more glucosinolates seem to start flowering later. But we don't see any effect of status. We also don't see any effect of fertilization, but you see a huge variation in all the data. And that's maybe also a very good trait, because being, again, chemical is also very diverse. So here we see quite a lot of variation. For example, in glucosinolates, again, may help you, because it's difficult for a herbivore to adapt to a plant if it tastes always differently. Yeah. So again, high chemical diversity may be a very good pre-adaptation to be well defended against herbivores. So to put that together for Bunyas orientalis, we have some typical traits of pre-adaptation. So what we found is that under fertilization, plants always grow better. Another pre-adaptation is this high chemical diversity. And then we have some traits that are more typical for post-introduction evolution. So at least the non-native plants invest more into bigger leaves, but invest less in defense then. And some data I didn't show you, but what we only found for the invasive populations, that under high nitrogen, they invested actually much more also than into the seed production, which also is also very efficient if you then want to spread in the invasive area. So again, we have many different mechanisms that help overall a plant then to become invasive. So to sum up that second part, First of all, these metabolic fingerprints may help us to really point to source populations. And then, having very distinct metabolites helps you to be efficiently defended against different kinds of herbivores, but also other antagonists like pathogens. And then finally, you have different pre-adaptations and different post-introduction evolution mechanisms that may all occur at the same time in parallel. So coming to the third and final part of my talk, contaminations, again a very different topic, but something I find also very important because we have more and more sites that are actually heavily contaminated, mostly due to mining activities. So within the last 100 to 150 years, these sites got heavily contaminated by heavy metals. And the heavy metals are the ones here in the period system. So if we focus in, zoom in, particularly zinc, for example, and cadmium. While they have different activities, zinc, for example, is actually in very low concentrations essential for us. Without zinc, our enzymes wouldn't function, and also plant enzymes wouldn't function. But if we have too high zinc levels, or have a plant has too high zinc levels, then it heavily disturbs iron homeostasis, so then it gets toxic. Cadmium is always terrible. There's also not minute amounts that could be beneficial. It's always toxic. It's toxic and carcinogen. It retards plant growth, and smokers know that they have twice as high levels in their body of cadmium as a non-smoker has. So it has really quite some toxic effects. But what is interesting is actually that there are some plant species that are able to cope very well with zinc and cadmium, for example, or other of these heavy metals, and they can hyperaccumulate these metals in very high concentrations, much more than all other plants. And these plants that can do it also belong to the Brassicaceae, also to Euphorbiaceae, and also to Asteraceae. So it's about 500 plant taxa that can do this hyperaccumulation. And I think it's a very interesting trait because this accumulation, there are some few sites where you have naturally high zinc and cadmium levels, but then a lot of these spots in worldwide only occur since human people start mining. So a colleague put it in the way that they said heavy metal tolerance in plants is probably an example of more powerful evolution in action than industrial melanization is in moth, meaning that, well, these sites, if you look at the sites, not much can grow there anymore, but few plants can grow there. And for example, one plant that I got interested in is Arabidopsis halleri. Arabidopsis halleri is a close relative to Arabidopsis thaliana. And if you look at Arabidopsis thaliana, you would find cadmium and zinc both in quite low concentrations. But if you look in Arabidopsis halleri, you see that the accumulation is about 1,000-fold higher. And they have these high concentrations of cadmium in their tissues, and of zinc it goes up to 10,000 microgram per gram dry weight, which is quite substantial. So they take up these compounds. They must have transporters that enable them to take up these compounds from the soil, and then they take them up into the above ground tissue. So the above ground tissue is full of these really toxic heavy metals, and you can imagine that that should also have a good defensive effect then on herbivores. So maybe, why do you invest that? Maybe to have also kind of an enemy-free space. So we tested that. We grew plants of Arabidopsis halleri on soil not amended or soil amended with zinc and cadmium, and then just scored in the field which herbivores would come. And over 3 months, scoring really every week for at least 2 days on the metal amended plants. Plants growing on metal amended soil, we found in total 2 herbivores over the 3 months for all plants that we looked at, whereas on the plants growing on the unamended soil that had then, of course, much less cadmium and zinc in their tissue, we found lots of different herbivores. And if you look at the biomass of the plants growing on zinc and cadmium containing soil looked also much more healthy. They produce more biomass. They have a nice green, whereas these ones show even kind of stress symptoms if they don't get enough zinc and cadmium. So it seems like they're even really well adapted already to these sites. Well, I told you in the beginning that we are getting more and more interested also in aphids. So the question was, how much of the cadmium and zinc is actually present in the phloem? Because, somehow it needs to be transported into the above-ground tissue, so it should be transported also partly in the phloem, not only in the xylem. And so we just placed aphids, a generalist aphid, on plants growing on unamended soil, metal-amended soil, and what you see is the aphids. So in the beginning we placed again 20 aphids on the plants, and after 3 days aphids survive quite happily on the plants without zinc and cadmium, but they die pretty soon, and after 5 days they're basically all dead. On plants where cadmium and zinc is available in the soil. And that indicates already that, obviously that's also a good elemental defense against aphids. So we wanted to know whether that is really due to the metals, because it could be also other traits that differ in the phloem. So we did some artificial diet experiments, added the metal, and also added the chelator to which the metal is attached in the phloem, with which it is transported through the phloem. When we did that with zinc and its chelator, then it actually had a positive effect on the aphids. And for aphids, zinc is also an essential micronutrient. It depends, of course, also on the concentration. If we would increase zinc a lot, then it may be also toxic. But at that concentrations with which we predicted that they should occur in the phloem, it was actually quite positive for the aphids. Cadmium was negative, so the aphids showed a higher mortality. But when we now combined zinc and cadmium, it was completely terrible, so the populations died and didn't reproduce at all. And that is interesting because it shows that there's some kind of potentiation effect. So once you have cadmium and zinc together, even more cadmium is probably put into the enzymes, and that's then more detrimental. So elemental defense, as I said, is then really active also against aphids. But we are always interested not only in the effects of the plant on the insect, but also what does the insect do with the plant. And these are the final few slides I want to show you. So what we tested was now how much does the aphid feeding affect also the plant chemistry in the phloem tissue. So here are again the plants on the non-contaminated soil. Here are the plants on the contaminated soil. And you see cadmium levels are very low on these plants here. Check the scale. So here it's 0.2, here it's 2. So there is indeed really much more cadmium in the phloem once the plants grow on cadmium-containing soil. So these are the data for cadmium. But what was interesting and surprised us also, that actually due to aphid feeding on the plants, so these are not aphid infested, these are aphid infested. Once aphids infest the plants, the cadmium level even increases. So we have a significant effect of the aphid treatment. I mean, it's not really necessary, because if the cadmium kills anyway the aphid, why increase the levels even once an aphid feeds on you? That seems to be something that may going on because the plant has some other transport changes in the transport once aphids suck on the plants. And the same pattern actually we get also for zinc. So of course the zinc concentrations are higher in plants growing on zinc-containing soil, but there we also have an effect of the aphid treatment. So zinc even increases, the zinc levels increase due to the aphid infestation. And Arabidopsis halleri is also a brassicase, so it contains these glucosinolates. So we also looked at the glucosinolate pattern. This is again an NMDS, so chemical composition is more similar between these samples than, for example, between this and that sample. Here are all plants on the metal contaminated soil. Here are the control plants, and then we also discriminated between young and old plants, and you see also plant age makes actually a huge effect. So whenever you do metabolomics, you need to correct very much for plant age or leaf age. But what is important here is actually that the soil had a significant effect. So metal plants growing on metal contaminated soil have a little bit different glucosinolate composition, particularly with this one glucosinolate was enhanced in phloem due to metal amendment of the soil. But here the aphid treatment had no effect whatsoever, so you cannot even see it in that pattern because there was no pattern due to the aphids. So Again, not only herbivores are important, but also fungi are important. So we also infested plants of Arabidopsis halleri with a generalist fungus, Alternaria brassicae. And here, the setup is that we have them then in this milkshake cups that are quite convenient to grow plants with fungi in it. And here we were also interested in kind of trade-offs. So if a plant invests a lot of, or has already a lot of cadmium and zinc, does it also then invest in some phytoalexins, so compounds that it usually induces to fend off fungi? And one of these compounds is camalexin, which is usually only induced in plants once they get infested by fungi. So camalexin is very low in plants that are not damaged, so you cannot measure it basically. It's on the baseline. If you damage the plants mechanically, there is some camalexin induction, that also differed slightly between plants growing in normal soil or on metal-amended soil, but that's also negligible. When we had the fungi on the plants, then actually camalexin levels were much higher in plants growing in normal soil, but were not that much induced on plants growing on soil that had cadmium and zinc. That shows us that there are some trade-offs potentially. So once you have a lot of cadmium and zinc, you do not invest that much anymore also in additional antifungal compounds. So at least there's some evidence, not for the glucosinolate, camalexin, for these trade-offs. So to put that together, I think we can clearly see that cadmium and zinc hyperaccumulation is a very effective defense against both chewing, biting herbivores, but also phloem sucking herbivores. Aphid infestation itself enhances, for whatever reason, the metal concentrations in the phloem exudates, but glucosinolates, so secondary metabolites, are not affected by the aphids. And we also looked at phytohormones. And then we have, at least against, again, we need to consider really different types of antagonists. We should not only look at herbivores, we also should look at fungi, because here effects may be quite different. And at least here in this antifungal defenses, we've found some indications for trade-offs. So I think to put all that together for the drought experiments, I showed you that drought regime can be also very relevant, and mycorrhiza can help to cope a little bit better with drought. Then for the plant invasions, I think it's very important to really look at shifts also in plant chemistry, and chemodiversity may be really a very important trait here. And then for the contaminations, this metal contaminations of some soils really has a huge effect on the different plant antagonist interactions. And I mentioned so much that high chemical diversity is very important, and we were very lucky that we could now attract the grant that we just recently got, where we really want to study in a group of 10 different groups throughout Germany the ecology and evolution of intraspecific chemodiversity. So we are really focusing on chemical diversity within plants. And if you know some people that are looking for a PhD position, we still have some positions open there. So feel free to contact me, and I would be happy to fill it with some interested people that like this topic. So with this, I'd like to thank particularly Clemens Stolpe, who did his PhD on the heavy metal hyperaccumulation. I want to thank Caro, who did the experiments on the drought and the wheat, and Lisa, who did the nice, beautiful work on the plant invasions. And this morning I was talking a lot about Rabea's work. That's Rabea. So thank you very much for your attention, and I'm happy to answer questions you may have.
Questions? I have one. Go ahead.
I have a few questions actually. I really like your talk. Thank you very much, first of all. I'm more of a volatile organic compound person, so I don't understand really how do you go and do the chemical or metabolic fingerprints. How do you classify them? Because whenever I try to do this kind of analysis, I find out that I'm missing 20 of them, that I may have only 10, whereas we're talking about 3,000. So how can you target all of the chemical composition in the plant, or distinguish the different plants with different fingerprints, chemodiversity fingerprints, when you are not able, perhaps, to measure all the, how can you measure all of the compounds?
Yeah, so we don't measure all of them. You are very correct. I should have said that we only look at the polar compounds. Okay. Yeah, so we do, we lyophilize the plants to quench the metabolism that no enzymes can work anymore. And then we do methanolic extracts. So it's really focused on the polar compounds. And if we then would add even non-polar extractions, then we would even get more. But yeah, it's already complex enough. So we focus on them. So we don't have the entire fingerprint. We only have the polar fingerprint. That is right.
Okay, and following with that, sorry, you show one graph that you are showing different methanol extracts, right? 10%, 30%,
Yeah,
100%.
yeah.
And then you say
Different
that
fractions.
with different extracts, you get very different compounds.
Yeah, so it was a methanolic extract, and we took different fractions of that. So we used an HPLC to get the fractions of the extract, and we tested the different fractions of that extract. But what was your question?
Sorry, it might be that I don't get this fractionation thing. When I think about making a solution, the whole solution is equal. So if I take different, I don't understand how you get different compounds when you are talking about the same extract.
Yeah, so we did more than I showed. We did also a non-polar extract and a polar extract. Only the polar extract had activities on methanolic extract. And then you can apply this extract either on a column, on a chromatogram column, and there you can even discriminate between the very, very polar that are eluted already in water and the not that polar that are eluted in methanol. And that's what you can do either with a column on an HPLC, or there are also the solid phase extraction columns where you apply your methanolic extract, then you apply water, and then just the very polar compounds come, then you increase the amount of methanol, then the little bit less polar compounds come, and then you use 100% methanol, and then the very polar comes.
Like a different gradient of concentration of methanol.
Yes. No, it's because I was confused because you were saying that, well, with different extractions you get different compounds, and I was like, hold on, this is the same plant material, so how is this?
Yeah, you always, in this plant extracts you have thousands of metabolites, and then you try to separate them again in pieces, your extracts, to always get fractions of these extract. Okay.
So many thanks for the talk, very interesting. I liked all three parts. I guess the part that really find more fascinating is all these plants, these Arabidopsis growing so well, and incredibly polluted soil, contaminated soil, fantastic. Really is a fantastic scheme for evolution, like you said, in action, evolution in action. And so I guess a very small question, very point. I wonder whether you consider the possibility that the actions of the heavy metals is directly through action on the phloem. Is it possible that the effectively these are equivalent to having extra potassium, say, in the phloem? Is it possible that having extra cadmium or extra zinc, in fact, increases phloem flow rates because it's an osmotic compound, it drives the pressure gradient into the plant?
So, that's a very good point. So it could be just a physiological aspect. That's why we also did, in addition, this artificial diet experiments to try to really separate. We also do not entirely separate it, because in the artificial diet you also then have maybe changed a little bit the pressure, but this artificial diet is actually a very simple thing. So you have a plastic ring on which you spread some parafilm, and then you put the dilution on where you have amino acids and sugars, so a typical phloem, and then you just add cadmium, and then you put another layer of parafilm in it that gives a pocket that has enough pressure so the aphid gets the pressure to be able to drink on that pocket. So there, I think then it's really the toxic effect of the cadmium and the zinc itself rather than any changes in pressure, because we cannot, the pressure is probably more or less just determined by the pressure we have in this parafilm pocket and nothing more.
I guess what the point I was trying to make, I understand this feedback on the aphids, but my point was the fact that the plants are growing fast. Is it can it just be a direct physiological effect. Because after all, if they are growing fast, it means that there is not they can handle the high concentration effect, the negative effect of it. But on the other hand, growing fast probably means having high photosynthetic rates. If you have high photosynthetic rates, you must have high fluid transport rates. I wonder, is that because they can actually use the contaminants to drive the transport?
Yeah, well, I mean, they're not really growing particularly fast. No. They just grew better than the ones without the heavy metals, but they are all rather slow-growing plants, so I don't think that it really pushes their growth in any way. They must have a very good tolerance mechanism because, I mean, also for a plant, cadmium and zinc should be super toxic, so they have some chelators to which they bind these compounds. I would doubt that it really helps, but I can't say for sure, of course. It's an interesting thought, yeah.
Yeah, Caroline, going in the same line about mechanism. I wanted to ask you about the mechanism that explains that when you have aphids, you have increased concentrations of zinc, for example.
Yeah.
But we can talk about that later. Even a more important question to me is, how do you use ecometabolomics to say something else to this field? Because at the end, after you talk, I wonder what do we really win with the metabolomics approach? I ask you because we work a lot in metabolomics, and I would like you to shed light in our approach, because at the end, what you are explaining here is more of let's say, classical analysis. So, are you giving a step forwards in understanding of chemical ecology by using metabolomics?
Yeah.
That will solve many of our questions, because we work a lot on metabolomics.
Yeah, so I don't just think that ecometabolomics can save the world, definitely not. But I think some new insights we got, for example, with this metabolic fingerprints, is that we can make at least predictions about invasion pathways, that you see some pattern that you wouldn't have been able to find.
I didn't talk much about that here. Maybe you didn't have time.
Yeah, we have more data. I couldn't show everything. And I think also for overall plant responses, I mean, just focusing on target compounds as we did before, you lack so much of the picture, So if you look at glucosinolates and you see to drought, they do not respond that much, but then you see, if you look at the overall metabolic fingerprint, the polar metabolites, not overall, then you see that actually a lot is going on, and you can discriminate between responsiveness of different species or different individuals even within a species. You get an idea how strong the responsiveness is, and also you get an idea how different species may actually respond to the same challenge. So if you challenge all plants, what we did, for example, we had 5 different plant species that we all challenged with the same mycorrhiza under very standardized conditions, and then we can also find, are there some metabolites that are changed in all plants in the same direction, which are more responsive, which are less responsive. In the end, it's only one step towards getting a better idea about other things. It doesn't tell us directly, but I think it makes you think about other hypotheses that you didn't have before.
That's very interesting. By the way, also when you work with different species, have you considered phylogenetic linkage?
That's something that we do now in the Drought project. We did it also in this mycorrhiza experiment that I just mentioned. There we had two species of the same genus, one species at least of the same family, and then 2 species of other families to get an idea. And of course, as you would expect, the closer they are phylogenetically, the more similar they are also chemically, and that you also see very nice along. And now in the Drought project, the colleagues from Spain have also, from Catalonia, have also different plants of the same family, at least. So we are there also want to look at these phylogenetic signals with the experts on that.
That's very interesting. Let me ask, the last question is about this drought, because here, as you know, many people we are working on drought, and then so the main compounds you talk about were proline, glutamic. So in your experiments?
In this experiment.
So what would you say that we should pay more attention to, these amino acids or sugars or secondary metabolites? You will say every
yes, I will say every compound.
But for example, most ecologists, we are not very well acquainted to, let's say, biochemistry. So a short summary of which are the key compounds that you would advise us to. So
if you want to have a key compound, I think proline is one of the key features. Some sugar alcohols give also a very good signal and a very reliable signal, but then for me I have to justify the metabolomics, and I think actually it's pretty nice if you look then also more in fingerprints that you see also different many different compounds responding to it, which we didn't account for before, because we always think just about the key compounds, but there's a lot also going on, of course, in the metabolism of many different compounds. I can't say particular groups, but if you just want to have the indicator, go for proline. That's also great.
Last question, now the truth, because then what do you do with these thousands of compounds that you are not able to elucidate? How do you work with them? And you see that they load heavily into the main axis for drought, for example.
So at the moment it's really trying to get over in the direction of pattern and look at similarities or differences, and also we can say how many are upregulated significantly or downregulated significantly, or increased or decreased, I should rather say. And then of course at some point we want to know the identities. So if we see that some compounds are always more going up or more going down, then of course we also try to identify those. But if we have 2,000 compounds, where would you start to identify? I mean, you can't do, because for each compound it takes a couple of hours to really get a good idea about the mass fractionation, and you would need to run separate experiments for that also. But then, I mean, at some point we get then back into detail and then also try to identify some that are coming up all the time, and that's then the second step that we do. But at the moment it's really looking mostly into pattern, but for some we get also identities or try to get identities.
Thank you very much.
Thank you for these good questions.
I would say about this classic chemical ecology and metabolomics. I think the approach we do as classic chemical ecologists, there is something wrong at the beginning. We choose some kind of compounds that we analyze them because we have the techniques, or maybe somebody at early analyzed those compounds and found those in the plants, and then it's easy for us just to apply a technique to our plant species, but even we don't know if these are the most important compounds. So I think that's a big change. Instead of focusing on the compounds, we focus on the plant, and doing classical chemical ecology, you may miss the most important responses, right? So, and I also agree that the identification part is the thing that needs to be solved. If we don't know what compounds are we talking about, we don't know about the biochemical relationship among them, and then it's hard to point what's the role of these compounds in the plant. So I think that's this should be the next step. I'm thinking bigger, but
totally.
And also, and even maybe a more bigger step would be to try to relate these compounds to a function, which in chemical ecology, I mean, many people talk about maybe anti-herbivore or chemical defenses. Maybe it's not a chemical defense, maybe it stimulates herbivory, maybe, I don't know. So this would be...
Yeah, but I think looking at this, just a short comment on that, because I mean, I tried to show you that different compounds can have also different functions because they act against different herbivores or different fungi or so. So that is already something that is an add-on that we see really they have different functions.
Yeah, so maybe we shouldn't stop assigning a role to a compound.
Yeah, like, but it has different roles for different...
They may have different roles at different times. So I wanted to ask, do you have any clue whether that in the native invasive study, that native population invasive population that was close to the native one, and you think that it's the origin of all the invasive populations, do you have any idea if that was the only population introduced or maybe other native populations were introduced and that was the only one that survived. Because I think the it gives a different type of information, right?
Yeah, I mean, it's hard to get this data really from the literature. We got excited that we found this one citation that this one population seemed to be the source population from where plants were introduced to UK. But often it's not very well documented, and to find the literature that really gives you more indications about different introduction pathways is hard. So I don't want to say that that's the way and that was the only way. It could have been also that actually there was some introduction back, because it's so different from all the other Asian populations. It could also have been that it was introduced to Europe, spread there, and at some point also somewhere introduced back. We don't know. I mean, that's nowhere documented in such detail. Yeah, it would be nice to know that better, but for Budleia, we have no better data. If any of you has, please give me the data.
And also my second question was about the specialist herbivore. How do you define a specialist? Because, I mean, it's an insect that feeds on one genus, one species?
Yeah, I mean, I didn't mean a monophygous species, but you mean when I discriminate between qualitative and quantitative defenses, I mean oligophagous insects, so that feed on few plant species within a plant family. Probably, I mean, there are very few that are really strictly monophagous, but let's say, for example, specialists that feed on Brassicaceae, they would be attracted by glucosinolates, whereas all other insects that are not adapted to Brassicaceae are deterred by glucosinolates. And glucosinolates I would consider as cheap defense, because they are easy to produce, there are not a lot of production costs. That the context how I meant it. But then digestibility reducers act against all herbivores.
Yeah, because I'm saying this because of the shifting defense. You don't expect specialist herbivores in the invasive place, but so in the model system I was studying before, Senecio, and there's these alkaloids, that are common in the entire genus. Mm-hmm. And you can find some insects feeding on many Senecio species, but Senecio is everywhere. So actually, if you define a specialist, like insects that feed on a plant genus, you find specialists everywhere. So
yeah, depending.
I mean, jump from one Senecio species to the other. Actually, this is what happens, and then you can find specialists and generalists.
But But interestingly, still you find less herbivory often in Brassicaceae. I mean, for example, Epidium draba is a Brassicaceae that is native to Europe, invasive in North America. And in North America, there are many other Brassicaceae, but still in a field survey, colleagues of us did found much less herbivory on Lepidium draba growing in North America compared to Europe. And you would think that, well, other Lepidium species are native to North America, and some herbivores should easily jump over to Lepidium draba, but for whatever reason they don't do. So maybe it's also that they just do not know that particular chemistry, because even though they have share glucosinolates, it's not the same composition, and they may have also, again, many other compounds that people didn't look at because we always focused on the glucosinolates. Yeah.
Okay. So, sorry. One question. Just out of curiosity, is anywhere working on fingerprinting insect-induced galls? Because it's one of the systems that the insect take control of how the plants grow. And that fingerprint could be really, really interesting in the insights of how that is controlled, because it's completely, well, at least it was completely unknown several years ago.
I'm not entirely sure. Maybe Renée Borges from India, from Bangalore, does, but I mean, she works a lot on galls and does amazing work on that. And I think she also went a little bit into the chemistry, but I'm not sure whether she does fingerprinting. So yeah, I'm not aware of, but that would be a really cool system to look at.
Okay, thank you.
Okay, so thanks very much again.